1 in 20 people will be diagnosed with colorectal cancer in their lifetime. What's even more concerning is that colorectal cancer is rising in younger adults, where cases in people under 50 have nearly doubled since the 90s. You may have wondered why we still cannot cure cancer despite the advancements in the medical field. Let me tell you why. Traditional treatments like chemotherapy damage cancer cells' DNA to prevent their cell division. However, cancer cells are really smart, somehow find a way to survive. When cancer cells become overexposed to drugs, they stop responding to them, which makes them resistant, a condition called drug resistance. If we can overcome drug resistance, we can cure cancer. Hi, I'm Deva. Here's where my project comes in. In my project, I developed tiny nanoparticles made of gold. Imagine nanoparticles as tiny vehicles delivering drugs specifically to cancer cells, avoiding the healthy ones. This helps to overcome the side effects observed in traditional treatments. Do you know that a single strand of human hair is approximately 100,000 nanometer in thickness, and my nanoparticles are 10,000 times smaller than this? Their main job is to stop the repair process in cancer cells. Let me explain. When there is DNA damage, any normal cell will activate the repair pathways, where dedicated repair proteins will assemble on the broken DNA site like the Avengers to repair the DNA damage. Here we want to stop the repair process because any cell with damaged DNA cannot divide or survive. To achieve this, I'm coating my gold nanoparticles with small proteins called peptides on their surface, which helps to stop the interaction and conversation with the repair proteins, and hence it stops the repair process. In my lab, we grow colon cancer cells. I perform an experiment called clonogenic survival assay, where I study whether the cells can survive after treatment. Here, I treat cancer cells with radiation to damage the DNA, and then I add gold nanoparticles to stop the repair process. And then I study whether the cells can still survive and divide after the treatment. If my treatment works, only few cells will survive. Out of 5 major repair pathways, I have targeted 2 pathways, and I observed at the highest concentration of radiation dose and nanoparticles, fewer than 10 cells were surviving. This shows that the nanoparticles have successfully entered the cancer cells, stopped the repair process, and hence stopped their cell division and growth. With this technology, we can overcome the drug resistance issue observed commonly across all cancer types. Additionally, these nanoparticles can be applied directly onto the tumor's surface through a simple endoscopy procedure. Now, remember when I told you about how my gold nanoparticles are coated with proteins? Both of them are non-toxic individually, hence the combination is also non-toxic. This treatment has no toxicity and fewer side effects to patients compared to traditional treatments. Every year, 44,000 people are being diagnosed with colorectal cancer in the UK. We all know at least someone close to us who has been diagnosed with it. Despite the failings of chemotherapy and evolving drug resistance, my research will develop an effective solution and save cancer patients' lives. Thank you.